Right, we'll start. Okay, we'll start with this, right? The first one today, it is almost the second unit. The first unit we have finished, left with probably the genetic and the physical mapping is left. I'll finish that in detail. I told you 2 or 3 classes. But today we'll talk about markers, right? Now, when you define a marker, what is a marker? Marker is nothing but a phenotypic or a genetic characteristic. Phenotypic or a genetic characteristic that can be easily genetic characteristic that differs among individuals. Otherwise, how will you find out and say that it is a marker? Characteristic that differs among individuals. That differs among individuals. Among individuals of a population. What else? It is not just enough if it differs among individuals. The next thing that is important is it should be, yes, it should be different among individuals and can be easily detected. And can be easily detected. Right? So when you say a marker, it can be a phenotypic marker. Let us say, like you talk about Ravi here standing and you have a black patch somewhere on my face. Okay. By using that black patch, you can say that, okay, this fellow is Ravi Kumar, right? Because he has a patch on the left side or the right side or that particular position. And similarly, if you look at your 10th standard mark list or something, they'll ask you the specific, the characteristics or the identification marks, right? And that can be easily detected. That's as far as the phenotypic markers are concerned. Easily detected in the population. Okay. Now, having said that, there's one more important term that is called as polymorphism. Most of the genetic guys must be knowing what polymorphism is. But you should understand that polymorphism is nothing but coexistence. You can write this particular definition in any way possible. I don't expect that you should write as it is what I'm writing over here. Coexistence of alleles. Sorry, multiple alleles. Write it as multiple alleles. That will be easy for us to. Coexistence of multiple alleles at a locus coexistence of multiple alleles at a locus in a population with a frequency of greater than 1% and what? And can segregate from generation to generation. Very easy, right? Nothing to mug up in this. Coexistence of multiple alleles at a locus in a population. At a particular locus, if you are looking at this coexistence with a frequency of greater than 1%. You call it a polymorphism only if you have the frequency greater than 1% and can segregate from generation to generation. Okay. Now, the classical example all of us know, it's the ABO blood group, right? For if you take a particular chromosome, if I'm talking about, see, always I represent a chromosome by a single line. And if this is the maternal one, right? And if this is the paternal one in an individual, like for me, like I'm an AB blood group, for example, I'll be having alleles at this particular locus as A and B. Some individual in the population can be a B group individual, can have a BB. B can also be what? B can also be a B and O, right? An individual in a population, some other individual in a population can have an O. So what is this? What is polymorphism? Basically, coexistence of these alleles that you have, A and B, right, and O, in the population, right, at the same locus. I've drawn the same chromosome. There will be some chromosome, right, which we designate as, let us say, some Z chromosome or Y chromosome or whatever it is, right, or some X and Y chromosome, right? And if you are, sorry guys, don't mistake me that the blood group is present on the sex chromosomes. No, I'm just referring to alphabets. Now, you have A and B and A, B and O. So you have A, B and O alleles that are present at the same locus and they are existing. So, however, the frequency of A, if you are talking about, should be greater than 1. 1% means 0.01. Means 1 out of 100 individuals measured should be having this particular allele. Right? Now, having said this, right, now you know what polymorphism is and what markers are, right? So you expect that, for example, if you are talking about phenotypic marker, I told you, right? Moles or any particular patch on the body, like which you can identify, that's fine. But here we are more concerned about genetic markers. So if you are talking about genetic markers, the next category that we are going to get into, right? There are two different distinct types of markers. They are called as anonymous markers, and the other one is candidate gene markers. Candidate gene markers. Now, what are anonymous markers and what are candidate gene markers? And why do you need these markers? We'll first have a look at that, and then we'll explain what anonymous markers are and what candidate gene markers are. For example, now let us say you must have heard about a word called as, popularly termed as QTL, right? This is more often than not used in genetics. It is nothing but a quantitative trait. What is it? It is nothing but a quantitative trait. Quantitative trait, right? It's nothing but a quantitative trait. Okay. Now, when you say quantitative trait loci, now you have to identify the difference between quantitative traits and qualitative traits. Now, if you talk about qualitative traits, you have discrete classifications for qualitative traits. Like let us say skin color. Skin color can be white, black, or brown. So you can easily distinguish between individuals that are black. So you have distinct groups if you can classify the individuals based on their color. So you have, let us say, this many number of individuals are white, this many number of individuals are black, and this many number of individuals are brown. So that's what is one of the discrete classes. Whereas when you are talking about quantitative traits, right, you may have, let us say, an individual. This is the quantity. Means like, let us say 1 kg of milk, right? And then you have certain number of individuals, 10 individuals producing 1 kg of milk. Then what happens? You may be having 1.00000001 kg of milk being produced by some other set of individuals. And similarly, 1.00002 again. So this makes this trait a continuous trait. Means for all the values that are possible, you can have this particular trait, right? Having values in a set of individuals across the individuals in a population. So that's how you differentiate between a quantitative, this is a qualitative trait, this is a qualitative trait, and this is a quantitative trait. This is how you distinguish between a quantitative and a qualitative trait. Now, having said that, fine. Normally, qualitative traits are governed by genes, right, that are specific to that particular quantitative trait. One gene alone, but you have one particular gene defining a qualitative trait, and these genes that define a qualitative trait are called as major genes. They're called as major genes. Now, what do you have? You have a qualitative trait, you have a quantitative trait that does contain, that's also again defined by genes, but this is defined by many genes. This is defined by a single gene, right? Whereas this is defined by many genes. And then, many genes, and all of these genes have got minor effects. So they are called as major genes and minor genes, or they have, all of them have got some effect on that particular rate of interest. Now, having said that, with this kind of a background that we already have for this, right, what we'll do is, right, now that you know what a quantitative trait is, we'll define what a quantitative trait loci is. See, quantitative trait loci is nothing but a segment on the chromosome. What is it? A segment on the chromosome, because you do not know how many genes would define a quantitative trait. So how are you going to tell them? Like, okay, this is a segment, right? Probably harboring genes. What is this? This is a segment that is probably harboring genes that explain the variation in the quantitative trait. So what do I mean? See, what I meant is, so the definition of quantitative trait is chromosomal segment. Chromosomal segment. Guys, I'm not giving you the recordings till I finish the entire this thing. No recordings anymore, because if you're not writing the notes, I'll not be giving any recordings. It's up to you now. Chromosomal segments. I want you guys to write down the notes as and when I teach, or if you're waiting for me to send me the recordings, there'll be no recordings from now on. Let me be very clear. Maybe that is one of the reasons probably some of the guys are not attending classes. Okay. At the end of this thing, probably once I finish the course, before your final exams, I'll send you the recordings. Chromosomal segments harboring genes that explain the variation of the trait. Means what? You have, if someone says, what's a QTL? It's nothing but a chromosomal segment, which you expect that particular segment contains genes, right, which explain the variation of that particular trait. So the most common trait that we are discussing is what? Milk yield. So if you are talking about milk yield, guys, milk yield is a trait, right, probably is governed by many genes, and ultimately what? You want a magic marker to find out, okay, if that marker is found, that genetic marker is found in that particular individual, that individual should be a high yielder. That's your ultimate aim, right? So now what people have done is, fine, now you know what QTLs are. QTLs are harboring genes. Now what happens? For example, if you have a gene, let us say a gene X here within this chromosomal segment. I'm again redrawing this particular chromosome. you are again harboring, let us say, you have a segment here. This segment has got a gene X here. This segment has got a gene X here, right? We are talking about some gene X. Now, if polymorphism is present within the gene, right? If polymorphism is present within this particular gene, and if this gene is influencing the physiology of expression of the trait, listen to me very carefully. If polymorphism means polymorphism is what? Presence of an allele, right? Now, when you talk about an allele, when you talk about polymorphism in terms of genes, if you talk about A, T, G, Cs, right? You have A, T, G, and Cs, right? Now, one individual may have a T here. The other individual in the population at the particular locus can have an AA. And if the frequency of this particular allele that we are talking about, of this single nucleotide polymorphism, which we are talking about, is greater than 1%, then you say that that particular allele is one of the alleles that can be considered to be existing in the population, defining the polymorphism at that particular locus in the population. So now, let us say you have a chromosomal segment defining different genes, having different genes that would probably control the expression of a trait. Now tell me, how would you find out the chromosomal segment? That's entirely a different story. You plan an experiment, you find out what are those chromosomal segments, right? And how to do it, we'll slowly discuss, right? Now just remember that we are just finding out, like, you have a QTL, chromosomal segment, harboring genes that could explain the expression of a trait. Now, having said that, now what we'll do is, you know that you have a gene X and there is polymorphism within the gene, polymorphism within this particular gene. Now, and if this particular gene is expressing the physiology of expression of the trait, then physiology of expression of the trait, then you say that this particular gene is a candidate gene. What is a candidate gene? Candidate gene is the one that explains the physiology of expression of the trait and harbors polymorphism within the gene, right? Within the gene, right? In the population, right? And which can be easily detected. Now, you have first, initially, what we have started with a marker, we have started with polymorphism, right? Now, slowly we are coming to genetic markers. Now, this is a candidate gene marker. Now, candidate gene is what? Candidate gene is the one that explains the physiology of expression of a trait. And what is candidate gene marker? If at all there is a marker in the sense like you have a single nucleotide chain or polymorphism within the candidate gene, which can explain the physiology of expression of a trait, then you say that it is the exact. Okay. Hopefully you've got this. Right. Now we have explained what? We have explained a QTL, we have explained. But, however, more often than not, you will not be finding out polymorphism within the candidate genes. Now, what happens if you have a QTL, right? You would like to, when you don't have polymorphism within the genes of a particular candidate gene which is influencing a trait which you expect, if you don't have that polymorphism or you are not identified that, for example, like you must have heard about a DGAT gene that is quite popular, which controls fat content in milk. Now, for example, that is said to be polymorphic in all the exotic breeds, but it is not polymorphic in indigenous breeds. Now, what happens in situations like that, like when you don't have polymorphisms, you would like to identify the nearby segments, right? nearby segments that would actually get along with the QTL. Means if this segment is there in the particular individual, you expect this segment, this particular segment or the QTL to be always present. Is that clear? For that to be existing, what do you need? You need a very strong linkage between the marker. You need a very strong linkage between the marker here, right? For example, this is a marker, right? And since this is not present within the candidate gene, these markers are called as anonymous markers. They're called as anonymous markers. Now you can differentiate the difference between two different markers that we have discussed. We have discussed about anonymous markers and we have discussed about candidate gene markers. And anonymous markers are the markers that are very strongly linked to the QTL, whereas candidate gene markers are the markers that are actually present within the QTL of a particular, within the QTL that governs a particular gene of interest, a particular trait of interest, right? Okay. Now that you know, candidate gene marker is very clear, like within a gene you have it. Now we'll discuss, when you're talking about anonymous markers have to have a very strong linkage between this, because any recombination between these two, marker and the QTL, the marker will immediately, what happened, will disappear from the population. So you expect that the marker, the term that is used in genetics is that a marker should be in strong linkage disequilibrium. Strong linkage disequilibrium. All of you are there in the class, guys. I don't know what I'm teaching for, but if you are there in the class, that will be great. Give me a yes if you are there.
Yes, sir.
Only,
yes, sir.
Only
Yes,
one.
sir.
Good. Equilibrium. Right. So what are we talking about? We're talking about markers that should be in strong linkage disequilibrium. We have a separate section for linkage disequilibrium, which we are going to talk in detail. Okay, don't unnecessarily think about this. I'll explain about this in detail in one of the practical classes, right? With an example, so that will be very clear. So there should not be any problem in understanding what linkage disequilibrium is. Okay. Now, when we'll have to finish all the anonymous markers too. We'll continue the class if you're breaking after 8 minutes. So, anonymous markers. Now, under the category of anonymous markers, you must have heard about RFLP markers, RAPD markers, and then you have what mini satellite markers. Now you already know what mini satellite is and microsatellite is. And I've asked this question. I don't know how many of you have written that. Mini satellite is present in the telomeric region, most common, right? Whereas microsatellite is present in the centromeric region. And this is the microsatellite markers, right? And the last one is the AFLP markers. Last one is the AFLP markers, right? Clear? So now what we are going to do is, first we'll talk about RFLP markers. When we're talking about RFLP markers, I assume that all of you know it's restriction fragment length polymorphism. And if the genetics guys are there, genetics guys are very thorough with this, but for the sake of all the others, there are two variants of RFLP markers. One is simple RFLP, and the other one is PCR RFLP. PCR RFLP. Now, when you're talking about simple RFLP, what people used to initially do is, okay, first you should understand what advantage people have taken in utilizing RFLP as a technique to identify markers. Okay. For example, restriction enzymes, the 4 cutters, hexacutters, tetracutters, or right. So here you have a tetracutter and then a hexacutter, 6 cutter. Which cuts more often? The 4 cutter cuts more often than hexacutters. You can just have a look at the web if you want to find out why and how. I'm not going to explain that. Okay. No. Now, for example, you have a 4 cutter and a 6 cutter. If in a particular segment, right, in a gene, if you're running a gene like this, right, and you have this particular, this thing will draw a single line as we have drawn earlier to avoid confusion. This is an A. Now, our easiest possible restriction enzyme is what? GAA TTC, that is EcoR1. Okay, let us assume that this is GAA TTC. Now, in a second individual, right, the first individual, if you are using this and if you are cutting it, what happens? You have a segment here and you have a segment here that is cut after the RE digestion. So after RE digestion, what do you have? You have a specific segment. You have different segments, right, which cut exactly. Now, in this particular individual of interest. Now, for example, you take a second individual at a particular point, which ought to have a GAA TTC, it has a GAT TTC. Now, what happens? This particular individual, which ought to have an A here, now does not contain an A here. So this restriction enzyme cannot cut, the EcoR1 cannot cut this. Instead of having fragments here, we'll be left with a bigger fragment. So that is how initially what they have done. They have taken the entire DNA. They have taken the entire DNA and they cut the DNA with the restriction enzyme, RE enzyme, right? RE enzyme. And then they have run the DNA after cutting the RE enzyme onto the gel. They have run this. So when you run this, what happens? There'll be a big strip of DNA. There'll be a big strip of DNA that is normally seen. Okay. There'll be a big strip of DNA because when you actually take your DNA and shear that into pieces, you'll be having all kinds of fragments. You'll not be having segments that are distinguished in the gel because you'll be having all sizes of fragments. Now what they have done is, now for example, this particular polymorphism they have identified in particular gene A. Gene A, right? Now you have a gene and this polymorphism is identified in a gene, that is gene A, right? They have taken the probe of the gene. They have taken the probe of the gene. Probe is nothing but a complementary segment which can probably go and hybridize to your fragment. And they used to have radioactive, right, nucleotides. Right? The phosphorus used to contain the radioactive material, and then they used to actually do it initially. But now we are not following this technique, but you can just get into your heads that they were using radioactive material then to identify RFLP polymorphism. Now, they used to take this, then they used to allow this probe to hybridize. Now, let us say if the probe hybridizes at this particular point, right, it means that there are no two segments, but there is a single segment like this, right? Now, in an individual next to it, if you're running a gel, where there exists a restriction site, Now what happens? Again here, you will be having a smear, complete smear here. And this smear now if you are using the probe again, right, what happens? Just because it is cut into small pieces, you'll be having two segments here for the same gene. Now you understand the polymorphism. They used to identify this contained a single band, whereas now this contains a double band. And likewise, initially they used to identify cutting the entire DNA, running it onto the gel, and then using the radioactive technique to identify using the probe of a particular gene of interest, where you already know that this particular polymorphism exists. Right? So that was initially the polymorphism that was given by a guy called as Grodziger. In the year, I think it is 1975. Right? Grodziger in 1975.